everyone. Today we're looking at the fungi, another eukaryotic group, which sounds kind of like a boring group, but actually I'm going to try to prove to you that they are actually very, very important. So I mentioned right here, why do we even study them? Many fungi live symbiotically with plants. And one of the huge things that they're doing is actually connecting the root systems between different plants. So underneath the ground, which we can't even see, fungi are there connecting plants to one another, as well as helping them to absorb nutrients. So in the protists lesson, you would have heard me say that there are fungi like protists, and what really set them apart was they had external digestion. So one of the things that happens with these symbiotic fungi that I'm mentioning is they release chemicals around themselves to break down things in the soil. And if there's a plant root nearby, it sort of benefits by being able to pull in those nutrients. And while doing that, the plant is also supplying the fungi with essentially sugars, which the plant is making through photosynthesis. So we call that a symbiotic relationship. It's essentially a little bit of a back and forth. Both of the species are benefiting. So I would highly recommend watching that video that I posted, which is essentially how fungi made all life on Earth possible. It will explain much further this symbiosis idea, but very, very important heterotrophs the fungi represent. So at one time, fungi were actually classified as plants. However, once we knew more and more information about fungi, we realized that actually they're not very similar to plants at all. So I've done a little Venn diagram right here, and you can sort of read through it, but comparing plants and fungi. So the similarities are in the middle in green. So there's actually very few similarities. Both are what we call sessile, which means they are non-moving. They're planted in their location. Both are eukaryotes, which is that they have a nucleus, as we talked about before, and they have similar growth patterns. And really what I mean by that really general term is we find them growing oftentimes both in soil. However, plants are using photosynthesis to grow, while fungi are using digestion to grow. So they are heterotrophic. So really, really important differences. I'll also mention just really quickly here, all plants are multicellular, which we're going to talk about in the next lesson. Fungi actually can be unicellular or multicellular. So I'll show you some examples of the multicellular type. They form structures called mycelium and hyphae, which plants don't have. So plants is the typical stuff like our stems, our roots, our flowers, and so forth. Fungi have a totally different structure. So in red here, I've written in big bold letters, really fungi are unique from the other heterotrophs because they have digestion outside of their body. They release these, what we call enzymes, essentially chemicals that break down their food, and then they just suck in the smaller nutrients. If you think of other animals aren't able to do that. We need to actually digest internally, take food in, chemicals break it apart, and we reabsorb it into our bloodstream. So a bit more about that later on. I wanted to give you a couple of phyla, which are five sort of main groups of where we typically find fungi. Now, I'm going to just focus on sort of the takeaways from this because I'm sure many of us have seen fungi out and about. Like if you think of things like mushrooms, right? Those are an example of a fungi. But what I want to point out are a couple of like key terms from this table. So one is saprophytes. Saprophyte describes really anything that lives on or in things that are decaying. So many of the chytrids, which is this phyla, they are living on or in something that's dead. And what's causing that to decay is that these are actually, these fungi, these organisms are releasing enzymes, releasing chemicals, and allowing those chemicals to break down whatever that surface was. So the next one, which we see often, I'm sure, is the food molds. So things that we see more so in our kitchen. And it looks sort of like green sometimes white. So I use the example of strawberry mold because that's one that typically we do see mold growing on. So those actually, those fungi came from the soil and they're just sort of left on the surface. So even after washing our fruit and vegetables, we'll often see them get moldy after a week or so, or however long it is. And what they're doing there is those tiny, tiny mold cells, fungi cells, are releasing chemicals onto the strawberry, let's say. And what we're seeing when we see it get white and fuzzy is actually the growth of that fungi structure. So a little bit more about that afterward. The glomerulocytes, excuse me, I'll just mention again, they are the symbiotic ones. And what I'll ask you to do is to watch that video I was mentioning that I've linked in the notes. Really helping each other, which is the symbiotic part, to supply each other with nutrients. This is one of the most important ecological relationships that we have. We have actually also, which you'll see in the next, in the lesson 4 that we just did, we have also some bacteria that are able to do that, but definitely the fungi are very, very important for cycling that. The next one, which I know some of you have been spending time baking over this quarantine, the next one are things like yeast. And yeast is actually one that can be pathogenic. You might have heard of like yeast infections. But the yeast that we use for baking is just a single cell yeast. So if you've ever had dry yeast that you just buy from the grocery store, that yeast is essentially in like a dormant state. It's being dried out. And what you're doing by proofing it, which is mixing it with warm water, is you're actually allowing it to then start metabolizing again. So the yeast will start to actually take in oxygen and try to actually get started. And that's why you often add like a little bit of sugar in there also. So more about that type of metabolism is actually in grade 12. So we're not going to talk too much about it today. And last but not least, again, things like mushrooms. So they are also cycling nutrients, and I'm going to show you on the next page a little picture of the mushrooms and then explain further that mycelium kind of concept. So the physical characteristics, as I was mentioning, the actual sort of structure of the fungi are made of these little hyphae. And I just show in this diagram here, there's like a little circle around the hyphae. That's like 1 or 2 cells. So you'll see that there's like division between the cells. You'll also notice on this picture nuclei. And some of the cells actually are dikaryotic, di meaning 2, or having 2 nuclei. And the reason why that happens has to do with these septums. These septums are like little openings that allows things to freely pass through. So where you have like one hyphae and then many of them, we would call a mycelium. So that's the term that you see up here. And the goal of this, if you think about it from the symbiosis perspective, is we have these like long channels, almost like little highways that are running under the soil. And so what happens is essentially the nutrients that this this fungi might pick up, it could travel way over here to another plant possibly, and the plant could then take in those nutrients. So these mycelium are essentially making this little highway for nutrients to pass around through. And in the video you'll watch, you'll see that also some of the plants are communicating other things as well through them, which is really, really cool. So moving on to reproduction, I'm not going to dwell on this diagram too much, nor will I ask you about this diagram. I use it because I want to just indicate and sort of show different parts of the life cycle of a fungi. So fungi are unique in that they can either reproduce sexually or asexually. We've seen that before actually with the archaea and the bacteria, for example. The protists have that as well, but here we have just a different process. And so the reason I've put this picture on is to show throughout the fungi's life, it can have different amounts of essentially DNA throughout the life cycle. So I was mentioning before, they can be dikaryotic, which means 2 nuclei. And if you have 2 nuclei, you also have twice as much DNA. So I just mentioned that as sort of an example. Here you can see there's like 4 at one stage and so forth. But I want to mention a really important term that we're going to start using in genetics, and that's really why I've included this here, is diploid and haploid. So this refers to the set number of chromosomes. So a diploid cell has two sets. So you and I, we have two sets of chromosomes, 23 from our biological mother and 23 from our biological father. And together for humans, that's 46 for most humans. The haploid would be if I have half as much. So if I have only one set. And in humans, that only happens in our sex cells, which are called gametes. So our gamete cells have just one set, 23, with the goal to be that when two haploid cells come together, it forms a diploid cell, which then becomes the new organism. So we're going to talk a lot more about these in a couple of days when we get to talking about human chromosome number and so forth. It becomes really important because we're actually going to start looking at how do the egg and sperm cells actually develop, and how can they be different from one another. So it's a little quick intro, but I don't want to spend too much time dwelling on this particular diagram. I'm not going to test you on it. What I want the focus to take away from here is that throughout the life cycle, the cells can change chromosome numbers.